How can we study alternative splicing in single-cell RNA-seq data? So first of all, what is the biological question that we want to study? We want to study the regulatory process of RNA splicing. This is a step between transcription and translation where parts of the RNA sequence, called introns, are removed, and the remaining parts, called exons, are joined together to form a mature mRNA transcript. By skipping certain exons, or only including them partially, multiple mRNA transcripts can be formed and each of them can lead to a different protein. This allows cells to produce different proteins, even if they are all encoded in the same gene. This alternative splicing mechanism is really cell type specific and is in fact one of the mechanisms that are used to do cell type differentiation. Because of this cell type specific characteristics, we use single cell data to be able to research alternative splicing at the resolution of single cells. These single cells can be clustered to identify groups of cells with the same cell type. This can be performed using popular clustering methods like SEURAT and allows to compare splice patterns between the different cell types. Unfortunately, next to these big advantages of single cell data, there are also some downsides. Single-cell RNA sequencing data is very noisy because we sequence the RNA transcripts of a single cell. This means that we have a low amount of starting material and results in dropouts and uneven coverage, which obliviates robust splicing quantification. In order to deal with all this noise, we are developing a tool that is able to robustly quantify splicing variation in single cells. In order to do this, we start by constructing splice graphs to represent possible splice variation in a single gene. Every transcript can be represented as a path between the source and the sink. In this small example, we have two transcripts. The first one includes the pink exon and can be represented with this path. The second one does not include the pink exon and can be represented using this path instead. Using these splice graphs allows us to efficiently represent the variation and to easily spot the interesting parts of the graph where alternative splicing might happen. Every time there is a branch point in the graph, there are multiple options for splicing, and thus splice variants may occur. The next step is to use these graphs to quantify splicing for each of the cell types. We do this by computing a percent spliced in or PSI value for each exon and junction in the graph. The PSI value expresses the relative abundance of a junction or exon in all the transcripts of a particular gene. By combining the values for all cells within the same cluster, we can compare the splice patterns between the different cell types. However, a robust computation of these PSI values is not trivial. An initial estimation can be made by mapping reads onto the splice graph and counting the read depth per exon or junction. But due to uneven coverage, some of the computed PSI values will be inaccurate. To address this, we use the conservation of flow property. This is a property that makes sure that the dependencies between different parts of the graph are met. PSI values are expressed as a percentage between 0 and 100%, and we know that each transcript follows a path from source to sink. Therefore, we have a PSI value of 100% in the source and a PSI value of 100% in the sink. This PSI then has to flow throughout the graph and there can't be any leaks. At each exon, we can check if the flow is conserved by looking at the sum of the PSI values of its incoming junctions, and if this sum equals the exon PSI value, the incoming flow is conserved. Of course, we can do the same for the outgoing flow. As we see in this example, the flow is nicely conserved at the beginning of the graph, but there are some violations around the pink exon. We can use this flow information to correct for wrongly estimated PSI values, like the ones here. This will make the computations more robust, which will result in better characterization of splice variation in single cells. If you are interested in this topic, please feel free to contact me.